Okay, so today we will. So yesterday we touched upon the effect of hormones on carbohydrate metabolism, and during that talk I also invoked the idea of lipid as a major contributor for of diabetes pathogenesis. So now we will in this lecture focus on lipids, its metabolism, and its role in metabolic syndrome. So let me minimize this. Okay, yeah. So what are lipids? So lipids are organic molecules that are soluble in non-polar solvents, but do not dissolve in polar solvent, that is water. So they are hydrophobic molecules. They include fatty acids, triglycerides, structural lipids, and steroids. So as in terms of function, lipids have a lot of function in our body. One of the more pronounced function is what we know is the cell membrane synthesis, because the membrane of animal cells are made of lipids. So they are part of the they form structural integrity of the cells. At the same time, they are also precursors for hormone synthesis. So as I've told you in the previous lectures that out of 3 classes of hormones, one is the steroid hormones. So the precursor for that are sterols, which are lipids. Beside that, bile acid production also requires sterol, and lipids also work as signaling moieties. Okay, so there are processes in which you attach a lipid to a protein, or lipid themselves can act as secondary messengers. So that's how lipids are also involved in cell signaling. But most of all, the lipids have as the molecules have been evolved as a major mean to store and utilize energy. So why lipid are preferred as a storage depot? So we'll get into that. First of all, this is a revision of yesterday's talk also on catabolism and anabolism. So like the carbohydrate, there are a lot of anabolic and catabolic processes which govern lipid metabolism. So this arrow, the blue one, actually shows you the catabolic reaction. So the breakdown of fat from the food and then to a much more simpler form like fatty acids are all catabolic reactions of lipids, which ultimately increase the production of energy. But at the same time, lipids are also synthesized as a storage depot, and this process shown by green arrow is an anabolic reaction. So it's the interplay of anabolism and catabolism which is required for lipid metabolism. So why lipids as a storage depot? Why we prefer them? Why we store fat? So the reason is that on an average a 70 kilogram person stores around 1600 kilocalories of energy in the form of glycogen, 24,000 kilocalorie of energy in form of proteins, and a staggering 135,000 kilocalories of energy in form of triglycerols or stored fat. And a gram of fat has about 9 calories, while that of a carbohydrate or protein only gives 4 calories. So it makes sense to store energy more in form of lipids rather than protein and carbohydrate. So that is an evolutionary advantage we have in having lipids as a storage material. Now, when we talk about storage material, we need it when we want energy. So the best example is when you are starving. So this graph shows you the utilization of different macromolecules, lipid, carbohydrate, and proteins when an animal starves. This example is how the mice utilizes these energy depots during starvation. So what you can see here is that as the time goes on, there is a decrease in carbohydrate utilization. So the red one is this one. The red one shows the carbohydrate utilization. So initially glucose is used up, but as we fast, the carbohydrate depot goes down, which provides us energy. So then our body start using fat as a major energy fuel, and it keeps on using it for a long time. Proteins, as we know, are preserved because they are needed for cell function. And it's only under extreme starvation we go for protein. But anyways, using protein or carbohydrate doesn't give that much energy. So why not use the fat first? And so that's why fat is the major source of energy under starving condition or under stress condition. So the two type of so this graph, actually these two graphs shows you the levels of glucose and fatty acids during starvation, how they affected the plasma level of these two molecules. So what you see here is that as the days of starvation goes on, your glucose level goes down. Now we know that the brain needs energy in form of either glucose or what I've told you earlier in the second lecture, that ketone body is one of the major source for our brain. So our brain needs to function all the time during starvation. So even though glucose is going down, fats provide a very important source of energy, that is ketone body. And that's how when you starve, the ketone bodies goes up, and these are derived from fat oxidation. So that is also one of the reasons why brain prefers to use fat under starvation. So that's how it provides energy to your neurons when you are starving. So now you understand why fat metabolism is so important, because we require fat to survive under prolonged starvation. So when we eat food, especially a fat-rich food, how it is absorbed in our body and how it enters to the metabolic cycles. So this picture shows you the fate of the fat after we ingest a fatty diet. So fat in the food generally comes in three flavors. One is the tags with the thiacyl glycerol, it is cholesterol esters, and the phospholipids. So tags are the major source of energy. Cholesterol is basically for cholesterol, and phospholipids are basically for membrane integrity, and cholesterol also for homeostasis. Now, after eating the food, these three molecules are these three types of fats are acted upon by a group of enzymes known as lipases. So in the stomach and duodenum, gastric lipases and pancreatic lipases act upon tags to break down into monoacyl glycerol and fatty acid and glycerol. Now glycerol can act as a substrate for glucose synthesis, and that we are not going to deal with in this lecture. But the fate of the monoacyl glycerol and other long chain fatty acids are to be discussed in this lecture. So the breakdown of so the tags are broken down into monoacyl glycerol, cholesterol esters are broken down into cholesterol and fatty acid, and similarly phospholipids through the action of phospholipases are broken down into lysophospholipids or and fatty acids. So after this degradation of fat in the stomach, bile acid is secreted, and it helps in the emulsification of fat. And these micelles then enter the enterocyte, which are the intestinal cells. Now, all these long chain fatty acid and monoacyl glycerol, cholesterol are taken up by the enterocytes, and they are then again packed into a structure of lipids known as chylomicron. And the unique thing is that at this step there are also some proteins which are embedded in these chylomicrons. So these are known as the lipoproteins molecules. And finally, these chylomicrons are secreted into the lymph and the blood stream, and after that they are taken up by the other organs in our body. So the first organ we'll discuss here is the liver, and this this shows how a hepatocyte process fatty acids which are derived from the intestine. So after the chylomicron are packed by the intestinal cells, they go to the hepatocyte, where hepatocytes possess an enzyme known as tag lipase. So these are lipases. They are called lipases because these are enzymes which break down lipids into smaller lipids. So chylomicron remnants are broken down into glycerol and fatty acids. So they are basically tags which are broken down into fatty acids and glycerol. Now fatty acid then enter inside the hepatocytes. So there are specific transporters present in the hepatocyte membrane which takes the fatty acids and deliver them inside the hepatocyte cytoplasm. So after fatty acid goes inside, it is acted upon by an enzyme known as acyl coenzyme synthetase, which joins an acyl group to the fatty acids. So acyl group is helps in the activation of fatty acid, and through this it can be packed into lipid droplets known as tag droplets inside the hepatocytes. So we'll talk more about these lipid droplets in the later slides. But apart from going into the packaging part of the lipids, acyl coenzyme A can also go into the catabolic phase. So instead of this anabolic process, this can also go into catabolic phase, and hereby fats are basically oxidized to produce energy and to make ketone bodies. So the product of beta oxidation is acetyl coenzyme A, which in turn acts as a precursor for making more lipids. So it goes into the pathway which is known as de novo lipogenesis. More detail will be dealt later on. And then as soon as the lipogenic pathway is activated, it blocks the beta oxidation pathway. So there is a negative feedback. So because you don't want to make lipid and burn lipid at the same time, it will be a futile cycle. So if you're making lipid, it means you have a lot of nutrition. That's why you want to store lipid. So you don't want to burn lipid. Okay. So either of the two state will exist at any given time. So as soon as the lipogenesis pathway starts, it shuts down the beta oxidation pathway. And then again, fatty acid synthesis can make fatty acid, which can again go into this cycle. So it's a loop which goes on, but at any given time, one of the pathway, either catabolic or anabolic pathway, will predominate. Now the tags which are made in the ER reticulum can be either stored as lipid droplet, as I've told earlier, in the hepatocyte itself, or the hepatocyte can just throw them out into the blood again. So this is done by packaging them into VLDL particles. So it's very low density lipoproteins, which in turn go inside the blood, and then either again go to the cycle of lipid breakdown and entering the hepatocyte, or go to other organs like adipocyte for storage. Now, when we talk about beta oxidation, so this process I'll be dealing in much detail in this next slide. So what is beta oxidation? So beta oxidation is basically the degradation of fatty acids to produce acetyl coenzyme A, which can go into the tricarboxylic or Krebs cycle to produce ATP, and also produce acetyl coenzyme as a precursor for either ketogenesis, gluconeogenesis, or for lipogenesis itself. So this is a very important molecule. So you have to understand that acetyl coenzyme is a precursor for a lot of biochemical reaction in the cells. So fatty acids are oxidized inside two organelles basically. One is the peroxisomes and the other is mitochondria. But we won't go into the peroxisomal degradation because it is much upstream and it's very long chain fatty acids which are degraded. We'll focus more on the predominant pathway, which is called the mitochondrial oxidation pathway, also known as the beta oxidation pathway. So in the mitochondria, fatty acids cannot go directly inside the mitochondrial matrix. So they have to be activated first. So this activation step requires an enzyme known as acyl coenzyme A synthetase. So this joins an acyl coenzyme moiety to the fatty acids, and then there is a molecule known as carnitine, which is attached to it through the process of through the action of this enzyme, which is called CPT1. So carnitine palmitoyl transferase. So by making this carnitine conjugate to acylated fatty acids, it can then enter the intramembranous space now of the mitochondria. This acyl carnitine fatty acids then can be translocated to the mitochondrial matrix by another translocase known as carnitine acyl carnitine translocase, and once inside the mitochondrial matrix, it is again broken down to its original constituents, which are acyl coenzyme A and carnitine. So this acyl coenzyme A actually is the lipid, the medium chain or long chain lipids, which are acyl CoA conjugated. Now after this, they can go into the beta oxidation pathway. So I won't go into the exact details of the metabolic processes, but what I want you to understand that through the process series of this metabolic process, each step you remove 2 carbons, and during the process of removal you generate reducing equivalents. So FADH and NADH2 is generated, and acetyl coenzyme A is made, which can be used up in the Krebs cycle or in the ketogenesis to produce ketones. Now this is a much more elaborated sketch of this pathway. So through every cycle of beta oxidation, it's called beta because it acts on the beta carbon of the fatty acid chain. Okay, so to the every cycle, so it starts from this. You have a fatty acyl coenzyme A. So suppose your fat is 14 carbon long. So in each step of in each cycle of this beta oxidation, you lose 2 carbons. So ultimately, in the end, you'll be left with 2 carbon fatty acids. And during this process, you will generate NADH and FAD, FADH2. And these can be used for the electron transport chain reaction, which all of you know and you must have read about it. So it needs reducing equivalent, which are actually generated through beta oxidation. Now this acetyl coenzyme A, which is made here, let me use the laser. Okay, so this acetyl coenzyme A, which is produced by beta oxidation, then enters through this another biochemical reaction, which is called ketogenesis, and it turn produce beta hydroxybutyrate. So beta hydroxybutyrate is the ketone body. Now this ketone, although this pathway is derived from a catabolism of fat, but itself this works as a precursor for further catabolism in the brain. So beta hydroxybutyrate is taken up by our neurons, and in a process known as ketolysis, it is broken down and used as an energy source. Now, as a byproduct, acetone is also generated. Now, if you remember yesterday's class, I told you that in diabetics, the smell of the urine smells like acetone. This is because in severely type 1 diabetics is a huge increase in beta oxidation, because the liver cells cannot sense glucose, so they burn fat, and that's why there's an increased ketogenesis and the blood become acidic, and that's why people die in type 1 diabetes. So this is the acetone which I was talking. Let's say it smells like acetone. Okay, so I've told you that once the fatty acid get inside the liver cells, it can be either stored in form of lipid droplets or can be exported out. So VLDL is the major form of lipoprotein which is secreted by the liver, and it constitutes basically of triglycerides. So this color shows you the triglyceride. This is the phospholipid composition, cholesterol and free cholesterol. So you can see there's a lot of triglyceride initially in the chylomicron. So this is the molecules which comes to the liver first from the intestine, and later on it is broken down into different lipoprotein molecules. And LDL, as you all know, is the major culprit for atherosclerosis, and HDL is the good cholesterol. Okay, so let's move. Now we have talked about the catabolic processes in lipid metabolism. What about the anabolic process? So it's not that we get all our fats from our food. Our body has devised a mechanism to make fat by itself. This is called de novo lipogenesis. So this is done inside our liver cells, and what it does is that it uses acetyl coenzyme A. So you remember this molecule from the last slide. So acetyl coenzyme A generated from beta oxidation again go outside, because unlike beta oxidation which happens in the mitochondria, lipogenesis or the anabolic process of fatty acid metabolism occurs in the cytosol. So acetyl coenzyme A has to go out from the mitochondria. This is done by a transporter known as citrate. So acetyl coenzyme A and oxaloacetate combines to give a citrate, which goes which goes outside of the mitochondria into the cytosol, and then through a series of reactions helps in making fatty acids. So it makes fatty acids in the cytosol, and this can be either stored as stack or could be secreted out from the liver. So that's how fatty acids are made. So these are called neutral lipid. Now let's go to the cholesterol biosynthesis. There's another class of lipid, as you know. So this is also acted upon in the liver to make cholesterol from acetyl coenzyme. So this is again the precursor for both the lipid synthesis pathways. So cholesterol also is made through a pathway known as the mevalonate pathway, and this is the enzyme which is one of the most famous enzyme, and is the multi-million drug industry is revolving around this, is because its inhibitor is known as statins. So we all know that statins are used to regulate blood cholesterol levels. So this is the enzyme on which statins works. It's called HMG coenzyme reductase. So as soon as you stop this pathway, you block de novo cholesterol biosynthesis, and you reduce the risk of heart blockage. And cholesterol itself act as a precursor for a lot of other biochemical substrates like bile acids, steroid hormones, and vitamin D production. Okay, so this is why anabolic reactions are important, because you generate much more complex macromolecules which can be used in the organisms. Now, how is lipid catabolism and anabolism regulated by hormones? So again, the main hormone which regulate these pathways is insulin. So insulin, in response to high glucose, is a stimuli which tells you that you should store lipid. So inherently it is a anabolic, pro-anabolic hormone. So it will instruct the liver cells to store lipids, to make lipids. This is done by activating an enzyme known as mTOR. mTOR in turn activates a transcription factor, which is a master regulator of lipogenic genes. So it binds to the DNA promoter of lipogenic genes, and then instruct the liver cells to store fats. Other hormones like glucagon act as an antagonist to this, and they actually instruct the liver cells to degrade fat. So they are anti-anabolic hormones or pro-catabolic hormones. And the same way, insulin also regulates the synthesis of cholesterol through another AKT pathway, which instruct protein known as FOXO3, which in turn affects cholesterol levels in the liver. So this is how the anabolic and catabolic effects of insulin are executed on liver by metabolism. Now we have talked about liver. Let's now move to another important organ which is basically meant to store lipids. So liver is a more dynamic type of organ. It doesn't store lipid for long. It just uses it. It stores it for transiently for some time, but its main function is to either catabolize it or send it out in the body to be stored in this organ which is known as white adipose tissue. Now, white adipose tissue are lipid storing warehouse. So what you can see is that in the cell of white adipose tissue, if you see under a microscope, most of the cytosol is filled with lipids. So it's a little bit space left for the nucleus, and almost all the cells is filled with lipid. So that's why they are the major lipid depot in the body. So lipid is stored in form of triglyceride, just like in liver. And the source of triglyceride synthesis is either through the chylomicrons, which are produced by the intestine. So some of them go to the liver, some of them comes to the adipocyte, and also glucose. So this is how the insulin instruct the adipocyte to store more lipid. So as soon as the insulin act on these cells, it increase the translocation of this GLUT4 transporter, which takes up glucose, and through the process of lipogenesis start making lipids from the glucose and store it in the cytosol of the adipocytes. Whereas other hormones which are catabolic in nature, like adrenaline and glucagon, activates the cyclic AMP PKA pathway and helps in the lipolysis. So lipolysis is degradation of lipid droplets to release simpler lipids like diacylglycerol, monoacylglycerol, and fatty acids, and which can be taken up by liver or liver to produce energy. So this is how the different hormones act on adipocytes to regulate lipid synthesis and release. So in net, this is the crosstalk between two major organs of involved in lipid handling. So adipocytes and liver cells. So the hormones shown in green are the one which promote the process, like insulin and cortisol promote lipogenesis in the adipocytes, and norepinephrine, growth hormone, and cortisol inhibit this process of tag generation. Cortisol, GH are also involved in the in increased lipolysis of fats in the adipocyte, and thereby releasing these free fatty acids. So these are non-esterified fatty acids. So when we call it non-esterified, it means they are free from the lipid droplets. So the free fatty acids are generally esterified to make lipids, lipid droplets or tags. So lipolysis is the reverse of that, and you release non-essential, non-esterified fatty acid, which are taken up by the liver, and you can see they can be used for beta oxidation, or could be again esterified in the liver to produce VLDL particle. So this is how this crosstalk happens and lipid metabolism goes on in the body. Now, why it is important? So you remember I said non-esterified fatty acids. Now, why it is important to store lipid? Why cannot we have lipid, just free fatty acid floating in the cells? The reason is that because free fatty acids, if you remember yesterday's slide, they are precursor for very reactive fatty acid species known as ceramides. So when you have a lot of free fatty acids roaming in the organs, they tend to make ceramides, which in turn are cytotoxic in nature. So they cause a lot of problems in the cells, like ER stress, oxidative stress, mitochondrial damage, and thereby increasing the your predisposition to insulin resistance, pancreatic beta cell failure, and diabetic complication. That's why liver cells and adipose tissue, what they do is that as soon as they see free fatty acids, they either burn them or they store them. Of course, you cannot burn them in adipocytes, so you can just store them. Whereas liver has two options: either they can just utilize them in beta oxidation or store them as lipid droplets to reduce this process of lipotoxicity induced by free fatty acids. So this is an important concept which is very important in diseases such as atherosclerosis, beta cell failure, and more importantly in a very epidemic disease known as non-alcoholic fatty liver disease. And this is a disease I will be talking to you in these coming slides. Now, we all have heard about metabolic syndrome. So metabolic syndrome is basically a defect or a disorder in the cellular lipid handling program. So it encompasses both defect in the carbohydrate and lipid, but now we know that lipid may be the first hit, which is causing the initiation of metabolic syndrome in humans. So you can see over the evolution of mankind how we have progressed technologically. So this was thousands of years before the industrial revolution and the technology came, and then in the last 50 years humans have gone on the sofa set like this, and they are dreaming about McDonald's. They're eating a lot of fatty food and carbo rich food, and then slowly coming to this stage. So this process is known as metabolic syndrome, and is related to the changes in the lifestyle of the industrialized world. So why is metabolic syndrome a lipid centric problem? The reason is that because lipid is the basis of all the major problem which is happening in this disorder. So it's not one disease which is metabolic syndrome, but a collection of different diseases, and you may have two or three of or all of them, or you may have only two of them, but still you are in a stage of developing all of them probably through, because they are all interconnected. So diabetes, it's a problem with the lipid in the carbohydrate metabolism, but its precursor could be the lipid derangement. Then you have the cardiovascular diseases, which are basically because of increased cholesterol or lipid handling problem in the arteries. Obesity is the major culprit, which release most of the fats to liver and cardiovascular complications. NAFLD is one of the most recent of these, and it is called non-alcoholic because it is in most of the patients which have fatty liver nowadays do not have a prior history of high alcohol intake. So this is exactly the terminology itself is now being changed into MAFLD, which is more metabolic associated fatty liver disease. So it's now considered as a part of this metabolic syndrome. So let's focus on NAFLD, because this is the area in which I have also been working for a long time. It's quite a serious problem in the world and in our country itself. So what is NAFLD? So in general, our liver cells store around 5% of the mass as lipids. So but when this lipid droplets content increase more than 5% of the total mass of the liver, then it is called steatosis or fatty liver. Now, fatty liver by itself is not a problem. It's not a pathological condition. The reason is that because I've told you that liver cells try to quench all these free fatty acids and pack them into lipid droplets, because lipid droplets are generally inert in short term. So they do not cause any major problem in the cell functioning. So when you have a lot of free fatty acid coming into the liver cells, for example, consider early insulin resistance condition. So your insulin is not telling your adipocytes to store lipids. So whatever lipid you're eating or the high glucose you're taking is causing your adipocytes to not take up lipid but release lipid through lipolysis. And this lipid, which is generally going as free fatty acids, is causing stress in your liver cells. So liver cells are making lipid droplets. So that's why your liver cells become fatty. So insulin resistance is both a consequence and a cause of fatty liver. And so this condition can remain for a long time. Many of the people can just have fatty liver and won't have any problem. But in some portion of the people, they progress to a much more severe type of fatty liver, which is known as non-alcoholic steatohepatitis. So this is a stage when your liver cells cannot compensate with the increased fatty acid influx. So they store lipid, but it reaches its maximum. So now the stored lipid are also oxidized by increased oxidative stress. Now these generate much more toxic compounds and cause ER stress and mitochondrial damage in the liver, which causes inflammation and apoptosis in the hepatocytes. So this condition is known as NASH, which is associated with inflammation. And in some cases, patients may progress to even much more severe cases of cirrhosis and also end up into having hepatocellular carcinoma. And unfortunately, there is still no approved drug for treatment of non-alcoholic fatty liver disease. So the only way you can go back to normal liver is through exercise and lifestyle modification. But as you know, this is not very much possible for everyone. So there's an urgent need to develop therapies and drugs for the treatment of NAFLD. Now, why this is a very urgent need? Because it is projected by 2020, India will have the highest number of NAFLD patients, and the rise could be as much as 164% increase. So the huge amount of population will be at present itself. In a study in Lancet last year, they showed almost 32% of Indian population have non-alcoholic fatty liver disease. And this is NAFLD. It is not, they have not stratified whether they have NASH or cirrhosis due to NASH. But if the total number of NAFLD itself increases, you can expect that NASH proportion will also increase linearly. So this is a global epidemic, and it's much more of a concern to us as Indians to try to develop new drugs for this disease. So what could be a strategy to get rid of this lipid? Now, this slide shows you what are the different strategies the liver cell can employ to get rid of the lipid, and that's where we are focusing the drug therapy now in the world, how people are focusing on this. So you have this condition, you have free fatty acid coming from adipocytes, you have chylomicrons coming from the gut, and over that you have a lot of insulin coming from due to insulin resistance or hyperinsulinemia. So insulin is driving increased lipogenesis, because I've shown in the earlier slide, insulin increases the expression of lipogenic genes. So it is using glucose to make lipid inside the liver cells. Apart from that, increased flux of chylomicron from the dietary fat is coming inside the liver. At the same time, increased lipolysis in the adipocyte is contributing to this non-esterified fatty acid. Now, when the liver is exposed to this high amount of free fatty acids, it can go to the two ways. As I told earlier, it can esterify and make triglycerides, or use the beta oxidation pathway to make ketones and degrade these free fatty acids. Now, the problem is that if you store lipid droplet for very long, they in turn increase the secretion of VLDL, because triglyceride could be thrown out from the liver in form of VLDL. VLDL can be broken down into LDL and could be oxidized to form oxy LDL and could have cardiovascular complication. And at the same time, these triglycerides, if they are stored for a long time, they could be unregulated lipolysis, and free fatty acid in turn could be generated to form ceramide precursors, which in turn will cause insulin resistance. And this vicious cycle will go on, because the more lipid you have in the liver, the more sensitivity to insulin you will lose, and insulin secretion will increase, and it will cause more lipogenesis to happen. Now, the next thing which liver can do is definitely to increase beta oxidation, but that is also a problem, because you can only increase beta oxidation to a limit and not more than that, because huge increase in beta oxidation will increase ROS production, because ultimately your beta oxidation is feeding into your electron transport chain. So ROS will cause damage to the mitochondria, and it will cause apoptosis and inflammation in the liver cells. So we need a way in which we can regulate both the lipolysis pathway as well as control mitochondrial integrity in a scenario where there is an upsurge in beta oxidation. So first aim of, so this is what I'll be telling you is basically my own story, how I worked on this this problem of degrading lipid inside the liver cells in NAFLD. And the first approach which I took during my postdoc work was to somehow increase the lipolysis of lipid inside the liver cells. So this process, this triglyceride breakdown of triglyceride into free fatty acids, because then we can shuttle them into for beta oxidation. Because in NAFLD generally this process gets quite slow, lipids are not broken down and they just keep on accumulating in the liver. So how can we increase this process? This was the major aim. Okay, so there are two ways in the in the liver how a liver cell can break down triglyceride or the lipid droplet. One is the classical way, which is more prevalent in the adipocytes, and adipocytes have these classical lipases, which are basically proteins. They are inactivated because they are clamped by a lipase inhibitor, but there are few proteins in the lipid droplet when they're activated by a catabolic hormone like glucagon or thyroid hormone. They increase the activity of these lipases and they act on lipid droplet, release free fatty acids, which can go into the mitochondria to generate ATP. However, there was a in 2009 there was a discovery that apart from this classical pathway, there's a process known as autophagy, which involves the formation of structures known as autophagosomes. So autophagosomes are double membrane structures which chew up macromolecules, other macromolecules. So like mitochondria or glycogen breakdown is also done by autophagy. But before 2009, nobody knew about autophagy as a degrading material for degrading mechanism for lipids. So what you can see here is that there is a congregation of proteins known as ATG family proteins, and they form a double membrane structure just above the lipid droplet membrane. Then this membrane encloses, the autophagosome structure encloses, and they just like a piecemeal type of eating up, like the Pac-Man style. They chew up drop by drop lipids from the big lipid droplet and then take them into the lysosomes. Now, classical lipase works in the cytosol. So the lipases are all in the cytosol, and the process is in the cytosolic compartment. But for autophagy to degrade lipid, it goes into the lysosomes. Now lysosomes, we all know, are basically the degrading suicidal bag. So it's called suicidal bag because there are a lot of enzymes which burst open can kill the cells. So they have a lot of enzymes which include hydrolases, lipases, but they all are active in the acidic environment. So lysosomes are acidic inside because they have pumps which pump in H+ ion inside them. So in the acidic environment of lysosomes, there are specific lipases which degrade lipid, and the fate is same as the cytosolic lipase. You release free fatty acid, and which goes into the mitochondria to produce ATP. Now, this process of autophagy was first discovered by Belgian biochemist Christian de Duve, and he was the one who got Nobel Prize for the discovery of lysosomes. However, this process, the cellular process of autophagy, remained, we can say, people were not paying much attention to this process unless until Professor Yoshinori Oshumi, who was a cell biologist in Tokyo University in Japan, used yeast cell to understand the molecular mechanism of autophagy. So although autophagy was described by de Duve early on in the late 1960s, the mechanism was not at all clear how it happens. So he's the person who worked out the mechanism, and he got the Nobel Prize for working out autophagy mechanism in 2016. So these are two big names in this field. Okay, so this shows you a picture of how the catabolic autophagy process takes place. So as I've shown in the earlier slide, there's a phagophore formation. There's a double membrane structure which takes up cellular cargo, which could be anything from a bacteria, a virus, intercellular organelles, and of course lipids also. And then it takes up, they fuse with the lysosome, and lysosomal hydrolases then digest these, and the product is released back into the cytosol. Now autophagy has a lot of role in the cells. It's just not for degrading things, it is also for quality controls, so, and energy production. So in this case, when lipids are being degrading, they are helping first to remove the lipid droplets, which is called degrading cellular lipids, and at the same time it helps to produce free fatty acid for energy production. But at the same time, autophagy also has some quality control or housekeeping functions, which is to remove any damaged organelles. So it's like they are like brushing all the damaged things from the cells so that the cells are always clean. So this is how autophagy works. And as any other metabolic process, it is regulated by hormones. Insulin is the one which stops autophagy to happen because it's expected. Insulin is an anabolic hormone, so it will stop every catabolic reaction in the cells. An autophagy being a catabolic process, it is inhibited by insulin. And the same thing with amino acids, they also inhibit, because when the amino acids are there, you don't need to break down cellular organelles to produce amino acids from protein. So they are both negative regulator of autophagy, and these are some of the mechanisms which I think I won't go in detail in this lecture. But if anybody wants to understand more autophagy, they can contact us, because our lab is basically focused on autophagy research. So why autophagy is important to study in as a novel target and therapy for NAFLD. So there have been reports in the past that autophagy, cellular autophagy, had been defective in patients with NAFLD and in animal models of so they both animal studies, and this study which was published in Cell Death and Disease showed in humans also, human liver, autophagy was defective. So definitely we need ways to increase this autophagic process to get rid of lipid and to regress the NAFLD progression. So how to find out those ways? Now, being an endocrinologist, I always look to hormones when I try to find out cures for diseases. Now, as I have been working in the thyroid field since my PhD, when I was doing my PhD in SGPGI itself, and then I had a very much focus, and you can say my love for this hormone, which is called thyroid hormone. Now, thyroid hormone, if you remember the slides, the second lecture, it's a very important metabolic hormone. So it helps in growth and it increases metabolism. So it increases the and it's basically a catabolic hormone. So it helps in breaking down big molecules into smaller ones through catabolic reaction. And it is known that if a person is hypothyroid, they gain weight. If they are hyperthyroid, they lose weight. So definitely it has a very potent effect on lipid metabolism. However, although there were few studies which showed that there is an increased prevalence of hypothyroidism in NAFLD patient, the exact mechanism of high thyroid hormone could be playing role in lipolysis in the liver and the progression of NAFLD was not very clear. And so when I was doing my postdoc, I had a great interest to see whether we can repurpose thyroid hormone or thyroid hormone analogs as a therapy to degrade lipid in the liver and as a therapy to inhibit the progression of fatty liver disease. So for this, I focused on autophagy as a mean to do that. Now, there was nothing known about thyroid hormone and autophagy. So the first thing I wanted to know whether thyroid hormone could induce hepatic autophagy. So this is the immunofluorescence picture which shows you the autophagosome formation in response to thyroid hormone. So these are basically liver cells which are overexpressing thyroid hormone receptor. So thyroid hormone, again, if you remember previous classes, they act through the steroid family of receptors. So they're amines, but they work exactly as steroid hormones do. So they bind to deep receptor and in turn activate the expression of genes. So in this case, thyroid hormone was we found that it increases the expression of several autophagic genes in the cells and in turn increases the autophagy. So you can see the what I'll tell you how. Let me use the laser. Okay, so this is a control cell and these red dots are basically autophagosome tagged by a protein known as LC3. So LC3 is a marker protein for autophagy. So when the autophagy is initiated, LC3-1 is converted to LC3-2. So it's a lipidation of LC3-1 which makes LC3-2 and it appears as puncta in the cells rather than a homogeneous staining. So the more bigger and dense puncta you see is basically autophagosomes inside the cells. So definitely what you can see here is that T3 is inducing autophagosome formation in the cells. Now the next question was, is the autophagy induced by T3 eating up lipids? So how do we do that? So we stain cells with a lipid probe, which is a dye known as BODP. So this dye actually stains the triglycerides or the neutral lipids. And then you use an antibody against LC3 to see whether these two stains overlap. Overlap means if an autophagosome is eating up a lipid, in immunofluorescence you will see red and green overlapping with each other, which will look as yellow. Okay, so red and green, when you overlay, they become yellow. So the more yellow signal you see in an immunofluorescence experiment, the more is the indication that there is increased lipid uptake by your autophagosome. And indeed, this was the case with T3 treated cells, and we could see more and more lipid inside the autophagosomes. Now, as a gold standard to see autophagy, you have to do electron microscopy, and when you see an electron microscope image, this is a T3 treated mouse liver section, and you can see here increased triglycerides inside the lysosome. So lysosomes are single membrane structure, but the autophagosome themselves are double membrane. So this is the autophagosome inside a big lysosome containing triglyceride, and this is the autophagosome which is going inside the triglyceride cells to chew it. So these are very good EMs which show that T3 indeed increase the lipid degradation through the autophagy pathway. So finally, to nail this point, what you need is to show that if you lose autophagy, then your thyroid hormone can no longer increase lipid degradation. So how do you do that? So do that, you use a siRNA. So this is a ATG5 siRNA we use. So this is ATG5, as you know, is the ATG family of protein which are known to regulate autophagy. So if you knock down one of these proteins, you lose the cells to invoke autophagic machinery. So in this Western blot, what you see is there that when you use a ATG5 siRNA, you inject it into the animal through tail vein, you lose ATG5, and in turn you lose your LC32 levels. So LC32 is the indicator of autophagosome formation. So when you don't have LC32, it means your autophagy is going down. And when you do that, what you see is that you measure the ketone levels in the plasma after knocking down autophagy in the liver. So you see that in the control mice, there's a basal level of ketone. This is the beta hydroxybutyrate, which is a ketone body, and it basically reflects the rate of your beta oxidation taking place in the liver cells. So you add T3, your beta oxidation goes up because T3 is increasing autophagic degradation of lipid. But when you lose ATG5, then your T3 is no longer able to increase ketogenesis or beta oxidation. So this showed that thyroid hormone induced lipophagy to increase fat oxidation in the liver. Now, the next question was that if you are increasing lipolysis, this step, you must be increasing free fatty acid entry into the mitochondria. So you must be seeing increase in mitochondrial activity. So how can a hormone, thyroid hormone, increase mitochondrial activity and without causing cellular damage? So to understand this process, you have to understand that fatty acid oxidation increase ROS, which is true. Fatty oxidation will increase ROS, and if this goes on for a long time, you will end up into a defective mitochondria or a damaged mitochondria. So you have to have some caretaking mechanism, homeostatic mechanism, which can take care of this problem. So this is a oximetry assay in which I want to show you that over time, as you increase the time duration of T3 treatment or thyroid hormone treatment, you have a increase in respiration rate of the mitochondria. This is oxygen consumption rate in the mitochondria. So you see it is a linear rise. It doesn't drop. So the mitochondria are still fine. They are healthy. Even though the respiration is going on, they are fine. They can deal with the upsurge in the fatty acid oxidation. So is it that they are not generating ROS? It's not true. ROS is being indeed generated. So this is a Western blot showing the amount of ROS produced. So this is basically protein carbonyl detection using blotting to show you that more and more ROS is being generated and it is affecting mitochondrial proteins in the cells. And this is a specific FACS mediated assay of ROS generated from the mitochondria itself. It is used, the dye used is called MITOCHONDRIAL. So this ROS is not generated from the cytosol, but indeed through the mitochondrial activity itself. So the ROS is being generated by T3. So how is it possible that you have a lot of ROS and still you have a healthy metabolism going on? So this picture shows you this is a mighty mitochondria. It's saying that I can burn everything off, but ultimately what happens is that under normal condition the ROS will make it fatigued and it will drop out slowly, and then it will induce apoptosis. But suppose you have a mechanism which can take care of the ROS and the damaged mitochondria. In this case, we hypothesize it to be autophagy itself. So what autophagy is doing, it is doing a mitochondrial pruning. So it is during the process of metabolism, it is picking up all the fatigued mitochondria, all the damaged mitochondria, removing them, so that at any given time you have a good pool of only healthy mitochondria. So you don't have a damaged mitochondria lying in your cells which can induce apoptosis. And this is what the process known as mitophagy is. So just like lipophagy, in which you have lipid being degraded by your autophagosomes, mitophagy is a process in which damaged or injured mitochondria are removed efficiently by your autophagosome. The process is the same. You have a stress, just like upsurge in the beta oxidation of electron transport chain activity. Then the cells have mechanism to pick up all the damaged mitochondria. So there is all this mitochondria which are damaged gives an indication or signal as a telltale sign that we are damaged, and it's better you eat up us before we start increasing the release of apoptogenic proteins. So autophagosomes are very good. They pick these mitochondria, they degrade them, and the cell at any given time has a good healthy population of mitochondria. Now, how do you see it in the cell? How do you see mitophagy in the cell? So there are some probes available. These are basically plasmid probes which have two color signals, which are called GFP and RFP. Now these GFP and RFP are tagged to a mitochondrial protein promoter. So what do is that when you overexpress this plasmid containing these two probes, you generate a plasmid which will make a protein which will go into the mitochondria and give fluorescence of both GFP and RFP. But the logic here is that what happens when your mitochondria get inside the lysosome. Now the trick is that in the lysosome, in the acidic pH, acidic environment, GFP fluorescence is quenched. So whenever you have an acidic environment, your GFP doesn't fluoresce. So what fluoresces? Only the RFP signal. So if your mitophagy rate is increasing, you will have more red signal and less of green signal. So when you overlay red and green, you will have more red than yellow. Okay, because when the mitochondria is in cytosol, you will have both green and red. Okay, so that is what is being shown here. So this is the control cell transfected with this plasmid. It is showing mitochondria, which is also, you can see as green and red is the same thing. So you overlay, you get yellow because there is no mitophagy happening. But when you add thyroid hormone, which increasing beta oxidation and mitochondrial damage, it will increase your lysosomal resident mitochondria. So there will be some mitochondria which will be only red and not yellow. So these are the mitochondria which are now under lysosomal degradation process, and this was increased when thyroid hormone is added. So that is how you probe mitophagy in the cell system. Okay, so again you have to use EM to be sure that everything is there, because best is what you see. Because probes are all indirect measures, but it's best if you can see through your eyes in an EM that indeed there is a mitophagy happening. So this is a EM of a mouse hepatocyte treated with T3, and we can see here that this is a mitochondria. Everybody of you will recognize mitochondria, but what you can see here, difference between a mitochondria, this one and this one, is that this mitochondria is being engulfed by a double membrane electron dense structure, and this is autophagosome. So this is a very classic image of mitophagy which we got. Is you can see, and this mitochondria actually is swelled a little bit, so it is under stress, and that's why autophagosomes are right there to eat them up. Okay, so this is also a autophagosome containing mitochondria, and so this is how mitophagy is probed. Okay, so just for your interest, this slide I'll show you I'll see whether you can play this movie. So this shows you in a real time how LC3, which is a marker for autophagosomes, overlap with this protein known as optineurin. So optineurin is basically a receptor for LC3 in the mitochondria. So when the mitochondria is under stress, it will call autophagosomes to get attached to this mitochondrial receptor known as optineurin. So let me see if I can play this. So what you have to see is that this green signal overlapping with the red signal. So red is mitochondria, green is autophagosome. So just remember this, and you see this video. Now if you see this video, you can see the green thing, which is the autophagosome, is trying to envelop this red structure, which is the mitochondria. I'll play it again. And this is how real-time mitophagy is visualized using time-lapse microscopy. Okay. Okay, then move to the next slide. So what was the summary of my research? So what I could decipher from my studies was that TH increases hepatic lipid metabolism via parallel induction of lipophagy and mitophagy, and TH, thyroid hormone, reduces NAFLD in animal models via induction of autophagy mediated lipolysis and mitochondrial activity. So definitely thyroid hormone had promising effect in animal models of NAFLD. Now this was a very interesting thing. So based on my fundamental research, we could actually go to a clinical trial, in which this was a phase 2 clinical trial in which we gave patients having non-alcoholic fatty liver disease 4-month levothyroxine, which is a T4 therapy, and we wanted to see whether what we saw in animals is also reproducible in humans. So can we use thyroid hormone as an anti-NAFLD drug? Okay, so this is the result, and possibly I was the only basic scientist and all our surgeons, because this is a clinical trial. And the reason I was here only because of my research to show fundamentally that this hormone can reduce. So this actually also tells you that whenever you do a fundamental research, it can, you never know, it can anytime translate into a clinical trial. So you don't have to be disheartened that whatever research I am doing in animals or in cell culture, maybe it may never go into the clinic. Actually, it may go into the clinic sometime. And this is what translation research is. So that's how I pick up this idea from patient studies that hypothyroid patients have more NAFLD. I worked out the mechanism of thyroid hormone, whether it can be used to prevent the disease. And finally, I went back to the bedside to see whether we can use this data which we generate through fundamental research back into the clinic. So this is how the cycle of translation research happened. And actually, when we did this clinical trial, we could see that the intrahepatic lipid content in patients decreased, not a lot, because we have to understand that we cannot give a lot of thyroid hormone to the patients. It can have a tachycardic attack. So we were giving thyroid hormone only to a level that the patients are not thyrotoxic. So they were still under the normal range, but on the upper quartile. Okay, so there was a decrease in fat content upon administering levothyroxine, and we also found a decrease in insulin resistance. The data is not shown, but we also saw that the insulin resistance was also lower. And this study on fundamental research was also picked up by pharma companies, and this was a study which used a drug, selective thyroid hormone receptor beta agonist. So what the pharma companies know is that we cannot give thyroid hormone, so they made us a very specific selective thyroid hormone agonist which can act on the liver itself and doesn't have a side effect on the heart. And they did a clinical trial, and they found, and actually the phase 3 clinical trial results are also out, and it shows very good efficacy in reducing hepatic fat in NASH patients. And these are some emerging therapies for NAFLD, and this is how this lipid induced disease could be. Finally, we think that we could get around it by affecting the hormonal status. So thyroid hormone mimetics, PPAR agonists, FXR, these are nuclear receptor hormone receptor which are under clinical trials for relieving NAFLD stress. And at the same time, insulin sensitizers are also showing good efficacy, and lifestyle intervention always remain as the major way to deal with it. And also cholesterol lowering drugs, because cholesterol also plays an important role in NAFLD progression. So with this, I'll end my slide, and hope so you have realized and appreciated how much lipid plays important on normal physiology and pathophysiology. Okay, so with this, if there are any questions,